Imagine for a moment a young child with cystic fibrosis, struggling with every single breath. Now imagine a mother who in her quiet moments sits by her child's bedside, praying for a miracle. Now imagine the millions of others battling genetic disorders like sickle cell anemia, Huntington's disease, or hemophilia, conditions they didn't ask for, but ones they're forced to live with every single day. But you don't have to imagine, because about 60% of Americans will face a condition with a genetic component at some point in their lifetime. And globally, 70 to 80 million people suffer from a genetic disorder. For these individuals, gene editing isn't just a scientific breakthrough, it is hope. But here's the problem. Hope isn't enough. Research isn't moving fast enough, and our current efforts, like the NIH Somatic Gene Editing Consortium, are not sufficient. They've made progress, but with limited funding and narrow focus on technology development, we risk delaying these life-changing treatments. This talk isn't just about explaining gene editing, it's about making a case for action. We need greater investments, whether from the NIH, private biotech companies, or global health organizations, to take these experimental trials to real-world treatments. If we don't accelerate investment and regulation in this field, we're making a choice to let suffering continue, and that's not a future that we should accept. So let's talk about real impact. Right now, scientists are using CRISPR-Cas9, a groundbreaking gene editing tool that makes precise, targeted changes to DNA. The system uses a short RNA molecule called sgRNA to guide the Cas9 enzyme to a specific spot in the genome, where it cuts the DNA. The cell then repairs the break, allowing harmful mutations to be corrected. Take Victoria Gray, a sickle cell patient, as an example. Her disease was caused by a single mutation in the HBB gene, which leads to the production of abnormal hemoglobin, called HbS. This makes red blood cells stiff and misshapen, blocking blood flow and causing severe pain. Scientists at Vertex Pharmaceuticals and CRISPR Therapeutics tackled this by targeting the BCL11A, a gene that suppresses fetal hemoglobin called HbF and its production. By turning off BCL11A in her bone marrow stem cells, they reactivated HbF, which took over for the faulty adult hemoglobin and stopped her red blood cells from sickling. The results: since her treatment in 2019, Victoria Gray has been free from the severe pain crises that once ruled her life. And it's not just blood disorders. In another case, researchers used CRISPR to modify blood stem cells in a patient with HIV and acute lymphomatic leukemia, called ALL. They edited the CCR5 gene to include the delta 32 mutation, and the patient's leukemia actually went into remission. While this didn't fully cure HIV, it proved CRISPR could target complex diseases at the genetic level. These cases show what's possible, but right now, technology is only reaching a handful of patients. If we want to make gene editing available to the millions who need it, we must commit to expanding research, streamlining regulations, and securing funding. The NIH Somatic Gene Editing Consortium was a step in the right direction. With $190 million in funding, it has improved gene delivery and reduced off-target effects. But here's the reality. It's too slow. Clinical breakthroughs are lagging because of regulatory bottlenecks and fragmented research. It's also underfunded. While $190 million may seem like a lot, in the world of biotech, it's barely enough to fund a single late-stage clinical trial. And it also lacks commercial scaling. Government funding research is vital, but without private sector investment, we will not see widespread availability. Gene editing needs the same urgency and investment that vaccines received during the COVID-19 pandemic. The faster we act, the more lives we can save. We need stronger public-private partnerships and more streamlined regulation to make these treatments accessible faster. And of course, when you talk about editing human DNA, people get nervous. Could we introduce unintended mutations? Could this become a luxury for the rich, widening healthcare disparities? But let's separate fact from fear, and let's start with addressing off-target effects. Advancements in gene editing tools like prime editing and base editing have significantly reduced unintended mutations. Prime editing, in particular, uses a modified Cas9 to fuse a reverse transcriptase enzyme, allowing precise edits without double-branded streaks. And let's talk about cost barriers. Every medical breakthrough, like vaccines and insulins, were expensive at first, but prices drop as technology scales. And let's talk about this fear of so-called designer babies. We're not talking about germline editing here. Somatic gene editing refers and affects only the patient, not future generations. So to those who worry about misuse, let me remind you that just because you think a tool should be misused does not mean that we should all abandon it. And let me use fire as an example to this end. When it was first discovered, some feared it, but it warmed our homes, cooked our food, and propelled humanity forward. Under the supervision of scientists, ethicists, and policymakers, we can ensure it burns brightly for the good of humanity, not recklessly. And this isn't just about saving lives, it's about economics too. The global cost of genetic diseases exceeds $1 trillion annually, from hospital bills to lost productivity. Investing in gene editing isn't just moral, it's economically smart. And if we don't lead this, someone else will. The US, China, and EU are pouring billions into biotech. The question isn't if gene editing will shape the future, it's who will lead it. If we hesitate, we risk falling behind in the greatest medical revolution of our time. Somatic gene editing isn't about playing God, it's about correcting mutations that cause human suffering. The real ethical failure isn't moving too fast, it's moving too slow. So we stand at a crossroad. We can either embrace gene editing responsibly or let fear hold us back. Every day, patients wait for a cure that science already has the power to provide. So I ask you, what kind of future do we want to create? One where we turn our backs on progress, or one where we invest in a healthier, more just world? This talk, once again, isn't just about science, it's a call for action. We need to push for expanded research, encourage ethical oversight, and secure investment necessary to bring gene editing to those who need it most. The choice is ours, and it's not one between caution and recklessness, it's one between stagnation and progress. So let's make the right one. And let me end off here with a quote by Alan Kay. The best way to predict the future is to create it. Thank you.